Thanks to decades of research and several missions like Curiosity here, we know today that Mars used to be habitable in the beginning of its history. It had a dense atmosphere, a global northern ocean, rivers and lakes, and temperatures similar to Earth. But unfortunately, 4 billion years ago, it lost its magnetic field. And without this protection, the planet progressively turned into the freezing, irradiated desert we know today. However, there is a small window of habitability, and the big question is, did life appear before the planet lost its magnetic field? And did it leave any trace we can detect today? To answer these questions, we're sending rovers to the Red Planet. And both Perseverance and the Roussel and Franklin rovers have for main objective to detect traces of ancient life. Later in the presentation, I'll present you one type of these biosignatures we're looking for. But here, you can quickly see how important the detection is. So, in order to know what instrument to use on Mars, where to send them to have a maximum chance to find potential signatures, we need to understand how these clues can be degraded over time. To understand how biosignatures degrade on Mars, we use Earth as an analog. Here is an example of all the steps that could degrade these signatures between the organisms living in a paleobasin and now. First, the incorporation into the sediments without being degraded or eaten, then the higher pressures and temperatures experienced during diagenesis, followed by additional potential exposures to water, oxygen, or microorganisms via fractures. And for the scientists to be able to collect and study these samples, we need them to be closer to the surface, which once again brings a lot of possible ways to erase our traces of ancient life. But that is for Earth, and Mars has an additional powerful destructive agent. Indeed, because it's lost its magnetic field, the Martian surface is exposed to heavy radiation. The first type, and most abundant, are the solar energetic particles. They're absorbed in the very first few centimeters of the surface. The second type are galactic cosmic rays. They are less frequent, but much more powerful, and penetrate deeper under the surface, up to several meters down according to models. There, they will react with the soil and produce showers of secondary radiation, including gamma radiation. When we look at the depth where these rovers are drilling, we see that the cosmic rays could have an impact on the biosignatures we're looking for. So the main question becomes, are any biosignature left at this depth? And knowing that would help us determine where to send the rovers and what to expect. To take into account this radiation, part of my project is to irradiate different types of analog terrestrial samples that naturally preserved biomarkers. I'm focusing on cosmic rays because, as we just saw, they're the main ones at the depth where the rovers are drilling. And I use gamma rays as analog because it's one of the main degradation products when the cosmic rays hit the surface. For today's presentation, I will focus on shales samples. Shales are fossilized mud. We have the Enspel shale here on the left with the physical trace of a leaf, and the Messel shale on the right with a fish. And amongst a variety of biosignature, they're both containing typical lipid fossils that I will present in a few slides. Not only fossils from the fish and the leaves that you can see, but also from microbial sources that are not obviously visible on this picture. After a lot of sample preparation, here are they, ready to be irradiated. They have been packed, flame sealed under vacuum, and the bottom of the tube is facing the radiation source. And this is how they looked like after radiation. They were exposed to a dose equivalent to 15 million years on the Martian surface with gamma rays. And then we compared the molecular fossils content and quantities between the controls that haven't been irradiated and the irradiated samples. If we don't see any difference, it's a good sign for the rovers, and we can keep increasing the dose to higher levels of radiation to see where the trend is going. And if we observe a decrease in biomarkers content, our objective was to characterize the rate and compare it in different types of samples. I'm going to focus for this presentation on a specific type of biosignatures belonging to lipid biomarkers. The first types are hopanes. They are fossils of hopanols, which are membrane lipids found in prokaryotes. As you can see, during diagenesis we are losing functional groups, but we keep the diagnostic hydrocarbon skeleton. And this structure, if found on another planet, is complex enough to be diagnostic of life. We don't know any way to synthesize this abiotically. The second biomarker are alkanes. They can be degraded from fatty acids here, as we can see. In this case, a simple chain of carbon can be formed without life, so it's not that one molecule alone that will give us any information about past life, but the distribution of alkanes in a sample. If they're mostly long chains or short chains, or if they have a higher amount of even or odd carbon number, etc. And the rovers that we sent on Mars have the ability to detect these hydrocarbons and to answer our questions. But we didn't expect at all the results we got. First, we compared the total organic carbon in our samples, N-spell on the left, methyl on the right. The controls here are in blue and the irradiated sample in orange. The arrow bar here represents the natural variation of the sample, so we see that there is no significant change here. So far, it's a good sign for the rovers, but here is what we saw for the hydrocarbons. They increased. Here is only semi-quantitative data. This is why we don't have any error bars for natural variation, but at these levels we can be confident that the amount of hydrocarbon is almost doubling after radiation. So we decided to look more specifically in our targeted biomarkers. First, we looked at the linear alkanes. And same thing, they increased to even higher proportions. After seeing that, we were curious in their distribution, because as I mentioned before, the distribution is the actual signature here. Here are the results, with the size of the carbon chain on the x-axis. And we can see that for both samples, even if the quantities after radiation are larger, the distribution itself doesn't seem to change much. We don't suddenly have only large or only short carbon chains, for example. So, for alkanes, the signal is not only preserved, but increased after radiation. For hopane's biosignature, it is not as clear as it was for alkanes. Same thing again, our two samples, before and after radiation, and some of the hopanes we identified in the samples. Again, this is not precise quantification, so it's hard to make strong conclusions. But we see different trends. Some stay at the same similar abundances. Here, in N-spell, this one is decreasing by almost half. And here, these last two in methyls are doubling. We don't expect radiation to lead to the synthesis of any of these because they're very complex, but we have a few hypotheses for hopanes and overall for a hydrocarbon increase after radiation. Concerning hopanes, most of the variation we see could be natural variation. For this, we will do additional quantitative analysis. But there are a lot of other molecules in the samples that we don't see in our runs due to the methods and the instrument we used. One of them are refractory macromolecules like kerogen or asphaltenes. Here is an example of their very complex macrostructure, making them not extractable by our method or visible by our instruments. But under radiation, they could break down and liberate alkanes, hopanes, or a variety of other hydrocarbons. And even if they don't fully break down, radiation could liberate pockets of adsorbed or occluded hydrocarbons that we now can detect. For this hypothesis, we will irradiate standards of kerogen and see if anything is liberated. Our last hypothesis is that our very well-preserved sample still contain fresher organics, like fatty acids, that wouldn't be detected in the hydrocarbon phase because they're still polar. But they could be degraded into alkanes during exposure to gamma rays. We will here irradiate standards of fatty acid to see if any hydrocarbon is created. Here is another way to represent our hypothesis. The total organic carbon, here in the blue box, doesn't change after radiation. Then, a part of that organic carbon are the extractable hydrocarbon. that we see in our rounds. They double in size, as you can see the green box. They include the alkanes that were increased by a factor of 2 to almost 4, and the hopanes for which we need more analysis to be conclusive about quantification. Outside the box of extractable hydrocarbons, we have the macromolecules in gray that could free some hydrocarbons under radiation, or fatty acids in dark blue that could be degraded also into hydrocarbons. Or it could also be a complex mix of all these processes. These counterintuitive but very exciting results opened a whole new plan of work to understand what is happening to our biomarkers under radiation. First, we will analyze our samples with precise quantitative instruments to determine real trends. Then we will re-irradiate the same samples but at higher doses to see how the trend is evolving. The results you saw were for 15 million years, and as a comparison, Gale crater, where Curiosity is right now, has been irradiated for about 80 million years. As we mentioned before, we will also irradiate macromolecules and fatty acid standards to figure out if they can produce hydrocarbons. And lastly, in this presentation, we focused on quantifying biomarkers that were present in the controls, but another question would be to know if radiation could be creating new types of biomarkers that we could look for specifically on Mars. With this, I'd like to thank Georgetown University, the Johnson's Lab, and the radiation facility at NASA Goddard for irradiating our samples. Thank you very much for your attention, and I'd be happy to answer your questions.